So our next topic that we are going to discuss is recombinant DNA technology. Recombinant DNA technology uh, is a technology that is nowadays widely used for the purpose of genetic engineering. And genetic engineering, as you know, it is the engineering of the genetic elements, that is the DNA and RNA, for creating novel life forms or creating new products. And here, just like that of any other engineering, you need some tools for manipulation. Every engineer requires some tools for their engineering purpose or modification of the existing ones. In case of recombinant DNA technology or genetic engineering, the genetic engineer also requires some tools and techniques for manipulating the DNA or the genetic components. So, in this first session, we will be discussing the tools for cutting and joining of the DNA and the basic concepts. Before proceeding, I would like to familiarize you with some of the terms that is already aware to you. They are transformation, for example. That transformation in the pneumococcus experiment, for example, that is the direct introduction of naked DNA or uptake of the DNA by a cell is known as a transformation. So, introduction of naked DNA or DNA to a cell is a transformation. The classical example is that of the pneumococcus transformation. We already learned in the basics of molecular biology. And another one is transduction. Transduction is a viral mediated transfer of a DNA or genetic element. There is a generalized transduction and specialized transduction. That's also some of the basic concept that is already known to you. Now there is conjugation and recombination. This conjugation and recombination are some process of gene transfer or exchange of genetic material, so that results in some form of new combinations of genetic element development. So the natural process of these gene transmission, that is transformation, transduction, conjugation, recombination, all these processes were a natural process of a genetic exchange. So such natural processes were basically restricted by imprecision. Imprecision means it is not a directed method which results in a particular or specific product. So it's a random process. Any transformation event or transcription event or conjugation, recombination, all these are natural processes and they are restricted by imprecision because it is not a specific directed process or a controlled process. It's a random process, so the product is unpredictable. Then screening and selection is another problem with such type of natural process of transmission, gene transmission. For example, you want to isolate a particular product or a particular organism or a gene that is producing a specific product. For example, an antibiotic producer, or an organism that produces an antibiotic or a drug against a particular novel uh, bacteria or a virus. So you have a specific target. But such natural process of transformation, transduction, or conjugation or recombination, they will not result in a specific gene product that will produce that product. So in that case, you require a large scale screening and selection process so that from a large amount of possible recombinant products or natural products that is produced in the environment, you need to screen and select your concerned antibiotic or the drug producer. That's why, for example, you want to screen for an antibiotic producer, you will collect large amount of soil samples or some other samples from different locations. Then you will screen the entire sample for a possible antibiotic producers. And from the antibiotic producers, you will see whether that antibiotic is successful against your target organism. So that type of screening and selection is required when you are targeting such a um, natural gene transmission methods. But for a genetic engineer, this process of natural selection and natural recombination or natural gene transfer methods is a limiting factor because it is a time consuming and also it is very expensive. And also such natural processes were basically limited by sexual compatibility between the species. So these transformation or transfection or conjugation, that type of gene transfer is basically limited by limited within that particular species. So between species or between species, natural transfer of genetic material is not normally possible. So the possible genetic recombination or recombinant products that is produced by natural methods will be limited because of the sexual incompatibility between that species. So these are some of the concepts about the natural process of gene transfer. So these limitations of the natural process needs to be overcome by specific process of genetic engineering so that your desired product can be quickly produced without much delay, or it should be time consuming and time saving, and also it should be economical so that undesired product should not be produced. Then only it will become a successful recombinant product which is useful for an industrial application. So this is about natural process of gene transmission. Now the recombinant DNA technology. It's a recombinant DNA. Recombinant DNA technology produces a recombinant DNA molecule, which is a molecule, which is a vector molecule. Recombinant DNA molecule is a vector molecule. Example, it's a plasmid or a phage or a virus into which the desired DNA fragment has been inserted to enable its cloning in an appropriate host. So a recombinant DNA molecule, actually it's a vector into which the concerned desired DNA fragment which you want to express in some target that is inserted. So your desired DNA fragment is inserted in a vector that vector may be plasmid, phage, or virus. So that virus or phage or plasmid which is inserted with your desired DNA fragment is a recombinant DNA molecule, and the purpose of which is inserting this DNA fragment and subsequent cloning in an appropriate host. They are formed by laboratory methods of genetic recombination, such as molecular cloning, to bring together genetic material from multiple sources. For example, the insulin gene from humans may be incorporated into a plasmid, and then subsequently it is transferred into a host cell like E. coli, so that it will be expressed in that target. So we are using molecular cloning methods so that it will bring together the genetic materials from different sources and subsequently creating sequences that would not otherwise be found in the genome. Hello, is it clear?
Yes, sir.
Okay. Now, there comes the importance of restriction enzymes and ligases. If you want to manipulate such genes from different sources, then you require some tools. They are restriction enzymes and ligases. Restriction enzymes were the cutting tools for the DNA, whereas the ligases were used for joining. We'll see the details later. And a chimeric gene is another gene, or it's a gene with two with genes from combined from two different sources. A chimera is as you know, it's a mythical creature with just like Narasimha in our mythology. It's a creature that is combined with the from two different sources or two species is combined together, just like Narasimha or some other mythical creatures. So a chimera is having two different species combined together. So basically, recombinant DNAs were a chimeric gene because it is a combination of genes from two different sources. And the objective of this technology is to obtain large number of copies of specific DNA fragments. So basically, when you are going for a recombinant DNA technology, your purpose is to produce large number of specific DNA fragments. So your desired DNA fragment needs to be multiplied in large numbers. Maybe that's for cloning or sequencing or different purposes may be there. So you want to amplify that or multiply that particular DNA sequence of your interest into large number of copies. So that is one of the objective. And another objective is to recover large quantities of the protein produced by the gene. So your target may be the DNA amplification itself or the product produced or the protein product produced by that recombinant gene. So large quantities of protein produced by the gene may be your target. Or to integrate the gene in question into a chromosome of the target organism to express in that target organism. For example, maybe the case of the green fluorescent protein, which is transferred by the Agrobacterium tuberculosis into a plant, where the gene is finally inserted in the host cell chromosome after the transfer, and the organism itself manifests that particular feature. So you have various purposes in while doing such a recombinant DNA techniques. That may be the production of large number of copies of the DNA or large number of copies of or large quantities of the protein product that is produced by the DNA, or subsequent integration of this gene, target gene, into the host cell chromosome and subsequent expression or transformation of that host cell into a novel cell that produces that particular protein for subsequent generations. So this is the purpose of a recombinant DNA technology. Now there is a vector. As we mentioned, the recombinant DNA is the chimera which is inserted with your target gene or target sequence, or the vector which is inserted with the target sequence. So the vector is a self-replicating DNA molecule into which the desired DNA fragment of the DNA or the DNA fragment can be introduced to obtain large number of copies. And there are bacterial plasmids or DNA viruses, etc. All these are different vectors. Then the DNA insert, which is also known as the chimeric gene, because it combines the gene from two different sources. One is a viral or plasmid vector, and the other one is a target gene, which is procured from some higher order organisms, like plants or humans, whatever. And the vectors containing these DNA fragment to be cloned. So vectors, the chimeric gene, this DNA insert, actually it is the vectors containing the DNA fragments to be cloned. So once the vector is inserted with these DNA insert or DNA fragment, target gene, then it is a chimeric gene. The chimeric gene is a combination of portions of two or more coding sequences to produce new genes. So this chimera or this recombinant DNA is a combination of portions of two or more coding sequences from new genes. And the host is the host cell into which this DNA insert or the plasmid or the chimeric gene is subsequently inserted so that it will be expressed in that host cell. Then cloning and cloning. These are the two terms. Two other terms. That's a clone is the asexual progeny of a cell or an individual. So a clone is a asexual progeny of a cell or an individual, and the process of production of this clone is known as a cloning. So DNA or gene cloning is the transformation of the host with the chimeric vector and the cloning, subsequent asexual multiplication. So that is a DNA or gene cloning. DNA cloning or gene cloning is the term that is used to denote transformation of the host cell with the chimeric vector. So for example, the plasmid, which is incorporated with your target gene, is incorporated into a host E. coli cell. So that is the transformation of the E. coli. Then subsequently it is multiplied so that it is cloned, and that asexual progeny which is produced by the E. coli host, multiplication of the E. coli host, and that overall process is known as a gene cloning. So the gene cloning includes transformation of the host cell with the chimeric vector. So that means, first of all, you have to produce a chimeric vector. Chimeric vector is the plasmid or another virus which is incorporated with your target gene. So once the plasmid is incorporated with the target gene to be expressed, then that will be inserted into the host cell, like E. coli. So the E. coli is now transformed with the chimeric gene. Then subsequently, that E. coli or the transformed cell will be grown in some medium, and that multiplication of the E. coli, the asexual progeny of the E. coli that is produced, will produce multiple copies of the gene, target gene, and also it produces the concerned desired product. So this process of transformation of the host cell with the primary gene and subsequent cloning or asexual multiplication, progeny production, is known as a gene cloning. So there are some differences in the terms that is used in case of animals, for example, transgenic animals. The transformation in prokaryotes, basically that's the uptake of the normal DNA or naked DNA, just like that of the pneumococcus. But the transformation in animal cell is quite different from that of the prokaryotic transformation. The transformation in animals or animal cells is actually conversion of the normal cell into a transformed cancerous cell. Uncontrolled growth is the feature of that transformed cell in case of animals. So the transformation process of animal cells is not simple uptake of the DNA, but it is the transformation into the cancerous state. So the animal cell transformation and prokaryotic transformation are two different terms. Please note that. And transfection is a viral mediated transfer of the or infection of animal cells. That is transfection. Is it clear?
Yes, sir. Clear.
Oh, okay. Now the next topic is about restriction endonucleases, which are the major tools that is used for splicing or cleaving these DNA by the genetic engineers. So when we discuss about restriction endonucleases, we have to understand two different types of nucleases, that is the exonuclease and the endonuclease. Exonucleases, as you know, they are nucleases which cleaves from both ends, from extreme ends of the DNA. So exonucleases remove base from the ends, whereas endonucleases cleaves in between the strand. So it won't act at the ends. So these are the two types of nucleases, that's exonucleases and endonucleases. This is already known to you. So they cleave. And restriction endonucleases, they are another specific type of endonucleases. Usually, these restriction endonucleases were when I ask about why these restriction endonucleases were termed as restriction endonucleases. Usually, the students will say that it is restricted to some particular targets or something. So it is not like that. The restriction endonuclease or the term restriction of the endonucleases, restriction endonucleases, that comes from host restriction of the bacteriophages. Maybe you might have learned in your basic microbiology text the host restriction. So usually restriction endonucleases were recognizing specific sites or specific recognition sequences. That's true. But the term host restriction of the bacterial phages means a particular bacteria will have its own non-self recognition mechanism. For example, a new cell or a bacterial cell uptakes a DNA, an external DNA, by transformation, for example. So now the foreign DNA, which is available in the medium, is now transferred into the bacterial cell. The cell will have two options: either accept the DNA and use it for transformation and subsequent genetic change, or treat it as a foreign particle and including virus or something harmful to the cell, and lyse that incoming nucleotide or the DNA fragment and treat it as a foreign particle. So normally every host cell will have its own defense mechanisms. The self DNA will not be lysed, or it will not be treated or cleaved by the host cell machinery. Whereas the foreign DNA, which will be slightly different from the host cell DNA, will be identified and it will be cleaved by the host cell defense mechanism. And this phenomenon is known as a host restriction, which is based on methylation pattern of the host cells. As you know, while we learn the DNA replication process, you might have discussed the DNA methylation. The new DNA strands will not be methylated during the replication process, but after the repair mechanism is already over, so that the new strand, after the synthesis of the new strand, the proofreading mechanism will verify whether there is any errors. So if there is no error, then that newly synthesized DNA strand will be methylated at A residues and C residues within the restriction sites. So there are specific restriction sequences where the restriction enzymes will act, and at that point there will be A residues and C residues which will be methylated by the host cell methylases. So every bacterial cell will methylate its own DNA after some time of its production, and now the methylated DNA will be actually accepted by the host cell as its own DNA or self DNA, because the methylation pattern that is applied to that restriction sites in the host cell DNA, or the new DNA that is replicated and produced in the host cell, is applied by the same cell. So it will have the same restriction pattern and or the methylation pattern, and that methylation pattern will be recognized by the host cell restriction enzymes and it will not be cleaved. Whereas a foreign DNA, which is coming to the cell by the process of transformation, and that will have a different methylation pattern, because that DNA, foreign DNA, will be methylated by some other methylation enzymes which is present in some other cells. So this will be treated as a foreign one. The host cell will lyse or cleave that particular foreign DNA. So normally all viruses will not be successful, or phages, bacterial phages will not be successful in infecting every bacterial cells. Only the bacterial phages that is having the same methylation pattern as that of the host cell will be successful in infecting the host cell. Is it clear?
Yes, sir. Clear.
Okay. So that is about the host cell methylation. So the host cell restriction, that mechanism identifies the methylation pattern of the new incoming DNA, and it subsequently cleaves the foreign DNA if it doesn't have the same methylation pattern. The restriction enzymes, restriction endonucleases, were specific to that particular site because it follows the same methylation pattern. So based on this methylation pattern, because these restriction sites were specifically methylated to a particular host cell, one restriction enzyme which is effective in one species will not be effective in another species. So these restriction enzymes were basically it is effective in one particular species, whereas not in another one, because of these host cell restriction features. Now, Liederberg and Messels in 1964 postulated the presence of restriction enzymes. In 1968, the first restriction enzyme purified from E. coli, K12, by Messels and Yuan. So that is the first instance of isolation of a purified restriction enzyme. That's in 1968. That's from E. coli, K12. There are four different types of restriction enzymes, or for classes of, or for types of restriction enzymes. Type 1. Type 1 is a complex with many subunits. There is about 15 base pair recognition sequence, or a single recognition sequence of a 15 base pair length is present in case of a type 1 restriction site. And they cleave, the restriction enzyme cleave about 100 base pairs away from the TCA located within the R site. So there will have a restriction site, and that restriction site will have about 15 base pair long, And once the enzyme, restriction enzyme, identifies that restriction site, it will not cleave in between, but it will cleave at a position 1000 base pairs away, almost at least 1000 base pairs away. That position will be cleaved. So the restriction type 1 restriction enzymes will not cleave in between the restriction sequence, whereas it cleaves at a distant location, maybe 1000 base pair away. That is the cleavage site of the type 1. So echo K and echo B, these are some of the examples of type 1 restriction enzymes. So it recognizes the sequence and cleave at some other location, not in between. Type 2 is a stable enzyme and that cleaves within the restriction sequence or close to them. So the difference between type 1 and type 2 is that the type 2 is also a stable one and it cleaves in between, not at a distant location. And more than 350 restriction enzymes for about 100 sequences were identified. So specifically, there are 100 different sequences, restriction sites, and it will be recognized by 350 restriction enzymes. That means many enzymes will recognize the same sequence. More than 350 restriction enzymes were identified, recognizing 100 sequences, 100 restriction sequences. It means many recognize the same sequence. And also it requires Mg2 plus ions for cleavage. And example is Hint2 enzyme of Haemophilus influenzae. That is another one example of a type 2 restriction enzyme. And this is basically used for cloning. Then type 3 is intermediate of two subunits, intermediate with two subunits, and they cleave within the immediate vicinity because it is not in between the restriction site, but about 24 to 26 base pairs away. So the cleavage site is in the immediate vicinity. There are some examples like ECO P1, ECO P15, Hint F2, so Hint F3. So these are some of the examples of the restriction enzymes coming under the class and the type 3 category. Now there is a type 2S category, which is two different enzymes, but the recognition site is asymmetric. So that the cleavage on one side of the sequence at about 20 base pairs away. So two different enzymes will be there, but recognition site is their asymmetric site means okay, we'll see that in one of the symmetry we'll be discussing with another image later. So type 3 is another enzyme, another category of the enzyme, and where we have two different enzymes involved in that one. But recognition site is symmetric. The asymmetric sites will be discussed later. The cleavage on one side of the sequence at about 250 base pairs away is done. So the restriction site is not on in between, but about 20 base pairs away. Cleavage is done by the type 2S category of the enzyme. So here, when we come to the nomenclature, the first letter of the genus is written in capitals. For example, ECO R1 is one enzyme name. So it is obtained from the genus Escherichia. So the E of the Escherichia is written in capital. Then the species coli, that two letters of the coli, CO, is taken in small letters, smallest italics. Then the strain name RY13 is the strain of the E. coli from which it is isolated. So that R, echo R, is the strain name of the E. coli. And the order of identification of the bacterial, that enzyme, that is the first enzyme that is isolated from E. coli strain R. That is the echo R1. So the order of identification for the first enzyme that is identified will be marked as first, and the second will be marked as second. So this is the normal nomenclature pattern of the restriction enzymes. So usually the first letter of the genus is written in capitals. First two letters of the species is written in small italics. Then strain, type or name is written as subscripts. Then if coded by a plasmid, the plasmid name is written as a superscript. When more than one enzyme is produced, then Roman numerals 1, 2, 3, etc. is used based on discovery or order of discovery. So all restriction enzymes were usually prefixed with the R for restriction marking as a restriction enzyme. So RE echo R1. So that is the normal, or that is the standard nomenclature pattern. But nowadays, you can't expect this to be seen in textbooks, because echo R1 is normally written without R or the prefix R, and these italics also may be avoided. And basically, the nomenclature is nowadays, it is the actual nomenclature pattern, but nowadays you can see some of the textbooks which is mentioning the name of the enzymes without these R prefix or some other italics portions. So this is the standard procedure for nomenclature, but nowadays it is not strictly followed. That's what you have to note. Okay, here is the restriction sequence of one enzyme, and you can see that there is the palindromes with the rotational symmetry is usually involved in such restriction sites. Palindromes, as you know, Malayalam is a palindrome. You can read it to both sides in the same manner. So that's a palindrome. So a palindrome with rotational symmetry. Palindrome with rotational symmetry means the second half will be a mirror image of the first part. For example, you can see that the GAA TTC. Here, actually, the palindrome is a GAA AAG, where CTT TTC is the palindrome. But when the second half of the sequence is rotated by 180 degrees, then the second half will be in the second strand or the complementary strand. The palindrome, the second half of the palindrome will be on the complementary strand. So there is a rotational symmetry. Rotational symmetry is there in case of such a restriction sites. So usually restriction sites or recognition sequences were palindromes with the rotational symmetry. The base sequence of one strand of the DNA double helix is the mirror image of the second strand of the or second half of the complementary strand. Is it clear? Hello?
Yes.
So here we have some point of cleavage. For example, on one strand we will have a point of cleavage here. On the second hand strand, there is another point of cleavage. And the digestion with the echo R1 enzyme, you will have two fragments created. So this is a staggered cut, which results in protruding or cohesive or sticky ends. So you need to understand the nature of cuts that is created by the restriction enzyme. Okay, here this is a staggered cut. A blended cut means it will be a straight cut, and both the strands will be cut on the same position. Whereas if the strands were cut on different locations or different points, so that a sticky or protruding end is created on both strands, then that will be a staggered cut resulting in protruding or cohesive or sticky ends. There are some examples like echo R1 or NLA3. So these are some of the examples of such enzymes which creates protruding or cohesive or sticky ends. So here is another example, BAM-H1 cut. It creates a 5' overhanging or sticky ends. Whereas PST1, it creates 3' overhanging sticky ends. So which end of the DNA is actually protruding or extending? So based on the nature of the cuts, either 5' end can be the sticky one or the 3' end can be the sticky one. So for your specific purpose of genetic engineering, sometimes you may require a 3' protruding or sticky end, or sometimes you require a 5' sticky end. It's just like a carpenter creating some wooden pieces with some grooves, and in some other cases you may create a groove on the left side, or some other cases you may create a groove on the right side so that it will properly fit. So in this case, you can create a 5' sticky end or a 3' sticky end based using such a specific restriction enzymes. So 3' or 5' overhangs or sticky ends can be created by such a BAMH1 or PST1. So this is another one where SMA1, that's another enzyme which creates a blend and cut. So it creates a cut straightly on both strands at the same point so that it creates a blend end. Sometimes you require blend and ligation with the DNA fragments. So you don't require a sticky end at that point. So then you can use this tool. Then another term is isochisomer. Isochisomer is a restriction enzymes isolated from different organisms but recognize identical base sequences in DNA. Example, ASP 718 that is, and KPN 1. These are two different enzymes and these have identical restriction sites. And isosomers actually they are isolated from different organisms, but the sequence is same. The recognition sequence is same. So usually one enzyme is specifically isolated from a particular organism, and it will the target will be a specific one. But isosomers were recognition restriction enzymes isolated from different species or different organism and acting on the same sequence. So they are isosomers. Now, is it clear? Hello?
Yes, sir. Please.
Yes, sir.
Okay.
Clear.
Now we will see some of the modifications of these cuttons. How it is these cuttons can be used for manipulating your DNA so that it will be useful in genetic engineering. The first manipulation or first modification to the cutton is removal of the 5' phosphate group by alkaline phosphatase treatment. For example, in one of the previous cases, using one of the enzyme, we created a 5' sticky end. So that 5' sticky end, that 5' sticky end is now available for further cloning and joining. But for cloning purpose, sometimes you require unwanted joining because your ligation reaction or joining to the concerned DNA fragment is to be regulated at your will. You are not interested in a random joining with any random piece of the DNA instead of your DNA fragment. So in that case, while preparing the strands or cuttings, you will remove the 5' phosphate so that without your permission, this will not go and randomly bind with any other fragment. So for which you require your fragment to be treated with the alkaline phosphatase and 5' phosphate group is removed. So it will not further ligate with any other fragment without you placing that 5' phosphate back at that position. Okay, so it's actually a treatment which is given to such DNA strands so that unwanted or unnecessary binding with some other fragments or random binding with other fragments is not possible. So the removal of the 5' phosphate group by alkaline phosphatase treatment is one of the standard procedures so that to control your recombinant DNA production process. And another one, addition of a phosphate group to a 5' OH by T4 polynucleotide kinase. So if you remove the phosphate, then you need to to again add that 5' before using that one. So you can prevent further joining of the strand by 5' removal of the 5' phosphate. But if you want to use it again, you need to add it back. So for which you can use T4 polynucleotide kinase. So two enzymes, that is a alkaline phosphatase treatment for 5' phosphate removal and subsequent blocking of the remaining process. And T4 polynucleotide kinase for adding the 5' phosphate so that further process will be continued. Now, removal of the protruding ends by digestion with the S1 nuclease. For example, you created a sticky end by one of the enzyme, and now you want to trim the sticky end, or that protruding strand, or 3' or 5' strand needs to be cut and removed, and you need to create a blended one. So once the DNA strand is cut with a sticky end creation enzyme, one of the strand, maybe a 5' end, will be protruding. The enzyme S1 nuclease is the enzyme that is used to remove this single stranded portion, so that the strand will be again made a blended one. So that can be used with another blended ligation process. Whereas a sticky end cannot be joined with a blended one. So in that case, you need to remove the protruding or that sticky end portion or single stranded portion from the end of uh, the strands. So that will be done by the S1 nuclease treatment. Then filling of the protruding ends, and there are two possible. Anyway, you are looking for a blended ligation, that is both strands will be of the same length. So now you have a sticky end is in your hand. So before going for a blended ligation, you have two options: either remove this one or extend the shorter strand to the full length. So then it will become the blended, or again it will become a blended. Okay, so S1 nuclease is used for removing the single stranded portion, whereas the shorter strand can be extended to the full length by E. coli clinofragment, E. coli polymerase 1 clinofragment, and that can be again subsequently ligated with the T4 polynucleotide ligase. So that means the shorter strands can be extended to the full length and again made it blended by clinofragment and synthesis of the resistor strand or shorter strand to the full length by E. coli DNA polymerase 1 clinofragment. Then removal of the nucleotides from the 5' ends using exonuclease. So you can again, for example, the strand may be a longer one and you won't require that one. So in that case, you can remove some of the strands from the 5' end by an enzyme known as the exonuclease. Exonuclease can be used for removing some of the bases from the 5' end. And also you can use some enzymes like E. coli exonuclease 3 that can be used for removing some bases from the 3' end. So these two items, that is a 5' end exonuclease and 3' end exonuclease, both creates sticky ends. So you have a blended DNA strand in your hand and you want to remove some of the portions and make it a sticky end one. So in that case, either 5' exonuclease or 3' exonuclease can be used for trimming one of the strands so that this blended strand can be created as a sticky end one. So there are various possibilities: phosphate addition, phosphate removal, then single strand removal by S1 nuclease, single strand extension by clinofragment, then double stranded region can be trimmed at 5' end by 5' exonuclease, or you can create another sticky end at 3' end by 3' exonuclease. So there are other tools like trimming tools. Now, treatment of double-stranded DNA with exonuclease is BAL31. So, double-stranded DNA, entire double-stranded DNA can be trimmed and its length can be reduced. In the previous case, only one of the strands of the DNA is trimmed or removed in the 5' end and 3' end cases. Whereas in the case of BAL31, you can have the trimming of both the strand from the end, so that the entire strand will be shortened. Then synthesis of a single strand tails or protruding ends at 3' end by terminal polynucleotide transferase. For example, a poly A or poly T can be added to the end of the DNA strand, to one of the strand, the 3' end. You can add multiple A units or T units so that a tail of poly A or poly T can be added by terminal deoxynucleotide transferases. So these are various modifications which will alter the end portions of the strands after cutting with the DNA strands. So this is required, just like as I mentioned, a carpenter who is manipulating the wooden pieces. They by trimming and chipping the or modifying the ends of the wooden piece. And similar to that one, the DNA ends can be because you want to join them together with another target. So based on the target sequence or the structure of the target sequence, you can modify your ends by phosphate addition, phosphate removal, or strand extension, or strand or trimming of the strands, or trimming of both strands, or even extending the strands by adding poly A or poly T at one of the ends. Is it clear?
Yes, sir.
Okay, now there is another item, that is a linkers. Linkers or adapters are molecules that can be joined to the cuttings, or these are the linkers are short, chemically synthesized, self-complementary, double-stranded oligonucleotides, which contain one or more restriction sites within them. So here we have a blended DNA, and this is an echo R1 linker. So the echo R1 linker can be ligated to both ends of that target blended DNA. So once the linker is attached here, unmute you please. Okay. So here the linker molecules were actually chemically synthesized molecules that can be attached to both ends of the DNA. So after the concerned enzyme, for example, this is an echo R1 linker with echo R1 restriction site. After blended ligation of these with the target DNA, the echo R1 enzyme can be applied. So at both ends of the target or blended fragment, now we have echo R1 sticky ends that are now attached. So this can be ligated with any target which is claved or prepared with the echo R1. So these linker molecules are chemically synthesized to short sequences with one or more restriction sites, and that can be attached to the target DNA, and subsequent cleavage will result in the production of the DNA fragment with the concerned restriction site attached at both ends of the desired DNA fragment. So these molecules were known as linkers. Okay. Then there are adapters. Adapters are short, chemically synthesized DNA strands, and there's double-stranded DNA strands which can be used to link the ends of two DNA molecules which have different sequences at their end. When used in conjunction with the linkers or adapters or other adapter molecules, can be used. For example, in the previous case, here we are attaching the same linker on both ends. So now this is prepared, it can be ligated on any target. But for example, if you have two such sequences, two such target DNA, longer DNA, you want to join with the gene A to B. Gene A and gene B is to be joined together. There you require an adapter. The gene A is prepared with echo R1 and gene B is prepared with a BAM H1. So the sticky end which is present on one of the DNA is echo R1 end and another one is a BAM H1 end. For joining them together, just like you are joining two diameter pipes, 1.5 inch pipe and 2 inch pipe can be joined together by an adapter, in which one of the end will have a 1.5 inch hole and the other one will have a 2 inch hole. So this adapter molecule will sit in between so that both the pipes with the different dimensions can be joined together. That's the purpose of the adapter. Similarly, in this case, the adapter molecules will have two different ends, and that chemically synthesized adapter will have a pairing region. This pairing region will hold that strands together, whereas on both ends we'll have an echo R1 and a BAM H1 site, and after cleaving and preparing, this will pair with BAM H1 and this will pair with echo R1. So the two different strands with the two different restriction sites will be now joined together, just like two pipes of different sizes were joined together by an adapter. So this is about adapters. So the conversion adapters join cut ends of two different restriction enzymes. So they are known as conversion adapters. So now there is a ligases. Ligases are also known as they are synthetases, and any one of a class of about 15 enzymes that catalyze the reactions involving the conversion of chemical energy and providing a couple between the energy demanding synthetic process and energy yielding breakdown reactions. So these restriction enzymes actually they are a class of 50 enzymes, these ligases, and they catalyze the reactions involving the conversion of the chemical energy and coupling of these energy with the peptide synthetic process. Sorry, that DNA is not peptide, but what's called that strand joining process, phosphodiester bond formation process that is done by using these energy. And they catalyze the joining of two molecules, deriving the needed energy from the cleavage of the energy rich phosphate bond, and it is in many cases ATP. So this the energy from ATP is used by the ligases for creating the bond, and a ligase catalyzing the formation of a carbon oxygen bond between an amino acid and a transfer RNA is called a amino acid RNA ligase. So there is another case. Amino acid RNA ligase is not applicable in case of DNA ligation, whereas these ligases are a class of enzymes which derives energy from energy-rich molecules like ATP, and it creates a bond for joining together of two DNA strands. And such similar ligases for peptides were also known as amino acid RNA ligases. So both the concepts were same, where it derives the energy. The ligase derives the energy from energy-rich ATP molecules and creates a bond between the two molecules that DNA molecules or that needs to be joined together. So the carbon-nitrogen CN bonds are formed by the action of such enzymes as amide synthetases and peptide synthetases. So there are different types of synthetases. The ligases are a class of or a member of these type of synthetases. Synthetases were usually seen in case of peptide synthesis or joining of the peptides, etc. also. So the ligases, DNA ligases catalyze the formation of a phosphodiester bond between 5' phosphoryl and 3' OH groups of the fragments in the duplex form. So T4 phage ligase, most commonly used for Blendon ligation. So that is the ligase that is used for Blendon ligation, T4 phage ligase. The enzymes that is mediating annealing, sealing, and joining of the DNA fragments is a ligase. Then it forms a covalent bond between 5' phosphoryl and 3' OH groups of the fragments. So this is the role of the ligases. Okay, these are the tools that is used in case of genetic engineering. So now we discussed the restriction enzymes, the different classes of restriction enzymes, then the nomenclature of the restriction enzymes, then ligases, etc. Is there any questions regarding this?